Good morning. Um, my idea, of course, I really do not know your background, and I excuse if sometimes I will be a little bit too repetitive or telling about concepts that are more or less obvious for many of you. Uh, Essentially, I thought that it would be nice to have an overview about vision. And then I will come in the second half of my talk, explain a little bit why timing and why this may be important to understand perception, particularly visual perception, and the point of view of brain or neuronal mechanisms. Now, why vision? I think that we have a lot of interest in vision because we are very visual as a primate. But also, vision neuroscience contributed a lot with important concepts on brain mechanisms. And I will talk more or less fast of some of these concepts. If you think what is seeing, it becomes obvious that seeing is linked to the idea of viewing. This is a very important concept because we are not just representing passively an image, but we actually interact with the world in a visual manner. So, from the perspective of understanding vision as a process, there is a lot of action in seeing. And here I present a little child learning to act visually with the world. And you see immediately that, in fact, vision is always linked to action. And there is a very basic concept in understanding vision, is that we see or we look for objects. So it's very important to understand that vision is not a passive representation of a visual world, but vision is a process where, we interact visually with visual objects. And this is not only defined visually, but is also defined with our action upon the world. Here, a very nice example that probably all of you know. This is called scintillating grids. And this is nice because it shows that perception is linked to the act of seeing. When you move your eyes, you can see these dots in the intersections. This is a very complex phenomenon, cannot be explained only by retinal mechanism. It exemplifies very well the complexity of understanding vision. Now, if we go then to the level of neuronal mechanisms, of course, we start with the very concept of a spike, because this is, so to say, a very important, not unique, but a very important signal in the brain. Now, so most of our efforts to understand brain mechanism, in particular visual mechanism, is, of course, concentrated in counting spikes, because this is, of course, a very important signal in the brain and how the brain represents the world and how it interacts across the different networks. And a very important discovery is that you can represent features, or the brain can represent features by the response of the cells. And here an example of many trials in the visual cortex of the monkey. You see clearly that the activity is not stationary. When there is a appearance of a stimulus, activity increases, and you can see by an increase in the frequency of spikes. And this concept, of course, was very important also for the idea or the notion that you can use numbers, no spike in the spike, to represent features. And this was, of course, very important for the development, the conceptual development of the von Neumann machines. Now, a very important concept in vision neuroscience, I would say the most basic, is the notion of a receptive field. And essentially what we have here is the representation of a portion, an area of the visual field. And if you record from a neuron, for instance, in the retina or in the visual cortex, you can find a region in the visual field where you have a modulation of the activity of this particular neuron. So the main idea here is that this response represents a piece of space in the visual world. And this triggered a series of experiments leading to very important concepts of the idea that receptive fields represent features. And this is the way the visual system used to extract information from the visual world. And a very first contribution was just showing that receptive fields of retinal ganglion cells have a concentric organization, and they are sensitive to the position of light in relation to the receptive field. There are two main classes of cells, on and off cells, and the on cells, they fire if the light stimulus happen to be over the center of the receptive field with tonic or phasic responses. And the off cells, they are the opposite. If you have light in the periphery of the receptive field, and then the cell fires. So this is a way of representing luminance levels at the very first processing level in the visual system, that is the retina. It was discovered in the 60s, and this was an important concept, that there are two major classes of cells in the retina of the cat. One is called X cell and the other Y cell, and they have different spatial temporal properties. I'm not going into much detail, but here comes this very important concept that the retina does not represent just luminance. There is a lot of processing happening already at the first level, and the receptive fields, they could see as spatial temporal filters. And in this way, the visual system will extract particular information depending on the response characteristics of the cells. In particular, Y cells have a very large receptive fields, and they are very sensitive to motion. And the X cells in the cat, they have a very small receptive fields, and they are sensitive to detail. So if you have very high spatial frequency, we use, and you see this many times in my talk, these moving grids or gratings. And it was shown in the 60s that the X cells are very sensitive to fine gratings, and the Y cells to, coarse gratings. And these differences were very important then to the development, for the development of the concept of parallel processing. And here an example of, in fact, the scope of this paradigm to characterize responses in order to understand perception. And you see here clearly that there is a variation of contrast and variation of spatial frequency. And you see that we perceive this as this form, as this envelope here. So we are not very sensitive to low spatial frequencies. In the same way, we are not very sensitive to very high spatial frequencies. And it's interesting to observe that if you characterize the response of retinal ganglion cells, this is a seminal work of Enroth, Cougar, and Robson, you see more or less the same shape. So this was exactly the goal of all these studies, characterizing response property of the cells and trying then to link this with perceptual phenomena. So this was, in fact, the basis, so to say, of understanding visual mechanisms. And it has been for, I would say, 40, 50 years, until we start then thinking that perhaps there was more to think about responses than just characterizing the activity of single neurons. So this is the main picture. Different cells have different receptive fields, and they can extract different information of a visual scene. For instance, the Y cells will have, so to say, a map like this, and the high-frequency contours will be characterized by the X channel. In the monkey, just a little bit of detail, this is called the parasol, the P, the magno channel, and the midget channel, the M channel, for the high-frequency components. Now, if you take the brain of a monkey and flat it, and this has been done mostly in the 70s, you can map point to point, using this concept of receptive field, the representation of the visual world in the brain. And the first interesting aspect here is that the monkeys, as we are, very visual. So a big portion of the cortex, more than 2/3, is visual. It's not purely visual, but there is a lot of machinery dedicated to vision. And going a little bit in detail on these maps, this is a kind of a summary, and you see that if we use this as a representation of the visual field, you have in the many different areas in the visual system, there is a lot of complexity, a representation of the visual field. And this is also a very important concept, the idea of a visual area. And these many different areas may have many different functions in extracting information from the visual world. And now I will go quickly into some important notions of the characteristics of the processing of some of these areas, like V1, V2, the middle temporal area, and the area V4. But there was also clear reasons why thinking hierarchies. For instance, the receptive fields of V1 are very small. Receptive fields of the infratemporal cortex are big as the whole visual field. So there was this notion that if you look at the response property of the neurons at different levels, you see that there was an increase if you take this as so to say, your main program to describe responses and to try to understand why a cell responds. And I will show to you this notion then became extremely solid because they could demonstrate that the receptive field response properties increase if you go from the posterior to the anterior brain. So there was a kind of a gradient. And also the notion that you have basically two components. One is a feed-forward system that, of course, was, so to say, very clear if you think that the visual system represents an image. So and the feedback system that modulates this representation. So these are basic notions that we have a lot, I hope, to discuss, but also extremely important, extremely, how you say, seminal. Basically, these, that the receptive field complexity increase over the levels, and that they can represent features of the visual world. And this was, in fact, the seminal work of Hilbert and Wiesel when they described that in the visual cortex, that the cells can represent the orientation of the stimulus. It's interesting to know that, in fact, this was a kind of a surprise for them, because we go experimentalists love to track their traditions, and that they came from the lab of Steve Kuffler, and they are looking, in fact, for properties similar to the retina, because they have been used to describe response property of retinal ganglion cells. And it was a big surprise for them to discover that in the cortex of an anesthetized cat, cells were sensitive to the orientation of the stimulus. And it took a while, in fact, to discover this. But this notion was very important because it went to the very idea that cell properties can represent features. This was a 16 millimeters movie they have done at that time in the 60s. Basically, the setup was an anesthetized cat and recording the single unit, so they are very careful at that time of isolating cells with these big electrodes made of glass and tungsten. And that they could record then the spiking activity of a single cell and characterizing with held hand lamp, something that we still use it today. There were no computers at that time.
Deixa eu falar uma coisinha?
Yes.
One thing that I find interesting is why did this work become so famous and why did they get a Nobel Prize? When Vernon Mountcastle, who was much more famous than, they were trying to copy Mountcastle. Mountcastle was discovering very similar things in the somatosensory system, and they were emulating Mountcastle and said, let's try to do something like that in the visual system. But then Mountcastle didn't get the Nobel Prize. Most of you probably never heard of him. He's still alive. And these guys, they became super famous. They're the most famous guys in neuroscience currently. And I think it has a lot to do with the fact that we are so visual, and they were not only studying vision, but they were making movies of it. So they could do this demonstration here. So it could take them to, it could take the experiments to the meeting, to the conference. And I think that convinced a lot of people that this was very important. I know Dawn is going to be.
I'm going to speak only because I can tell that Siddhartha is hoping that I will be an asshole. I think that part of the answer to his question is that they saw something that is very simple to explain, something that was very compelling, and most of all, something that required zero analysis. The analysis that they're doing there on those responses is what we like to call the FOT, the fucking obvious test, where they're not actually doing any math, which I think we're going to find out through this week is something that leads them astray. It's also worth saying that they got the Nobel Prize because they weren't as aversive to talk to as Vernon Mountcastle always is.
They're not. So here, in fact, they propose a very simple model to explain the orientation selectivity in the cortex based on the connectivity from the LG and so the nucleus that receive projections from the retina. And here, perhaps a little bit difficult to understand because it goes into much detail. We have simple and complex cells. This is a complex cell in the cortex. And this is more recent study of Reid and Alonso, a very fine detailed study when they recorded simultaneously single cells in the LGN, so that projects to the visual cortex, and the visual cortex cells. You see here these slabs. These are a representation of the receptive field of a visual cortex cell with the subzones, excitatory, inhibitory, and they could demonstrate by directly recording from these neurons that the off cells project to, has a correspondence to these inhibitory regions of the receptive field, and in solid lines, the excitatory region is, so to say, innervated by on cells of the lateral geniculate cells. So this is a very fine explanation of the organization of simple cell in the cortex based on, so to say, the model of human Wieser. And I think that they were very, without computers or too much mathematics, they were very convincing and explaining a very important model in how the V1 is organized. And this was, as Siddhartha said, because they could also demonstrate, so they are very good in observing. They said, oh, when you go with the electrode, the properties tend to be the same if we go vertically. But if you go a little bit tangential, there is a systematic change in the orientation preference. So then they thought, oh, there is an order here, and they proposed what is called the ice cube model on the organization of representation, of orientation representation in the cortex. And this evolved also taking into account other properties, for instance, the ocular dominance. I'm not entering into much detail, just to give the notion that there is a lot of complexity, but also there is a lot of order, mainly in view 1. And they're taking this, in this film, Antonio Galvez loves this film. So this idea that you can now characterize mathematically this order. So there is a lot of patterns that emerge in the organization of the visual cortex. And this is a very nice example, an optical imaging of the cortex. So you can record luminance levels. They are very, how you say, tiny differences depending on the way you stimulate the cortex. So if you have a whole visual field grating with certain direction, you can then excite only the cells that are sensitive to this orientation and build these nice colored maps showing that in fact there is a lot of order, mainly in view 1. And this can be, so to say, the, a map to explain interactions, and then we can build, so to say, clear notions of how the visual system represents an image. Any question? This has a very nice singularity. Biologists, they love to, I think that the physicists also to point to, so to say, gems. And this is one, you see that there are what we call pinwheels, and that this is a singularity in the map, how it organizes. And you see that here all the cells have, so to say, many different orientations. And from the pinwheel, there is a clear order in the representation of the orientation in the visual cortex. So this is the first basic notion. In view 1, we have representation of a very simple feature. And in this notion that response properties represent features, this is a very important feature, the orientation and direction of movement. And this has been obtained, as I have shown to you, by this oversimplification of an image into a bar, and they have been very successful, so to say, in simplifying our visual world to a point that one bar suffices to show this important characteristic, that is the representation of orientation. Yes?
I said the connectivity is a dynamic thing, and by any training or any activity, any experience, it can change. So how can we interpret based on the connectivity which it is not static?
It's not true that connectivity is completely dynamic. For instance, the connectivity between the LGN and the cortex will be always there. What changes is effective connectivity. So you can change the balance of the synapses and the whole system will change. Of course, to explain this is not so simple. And I will come at the end of the talk of some of the possible mechanisms that could explain this. So I'm not saying that everything is explained in terms of fixed connectivity, but a lot can be explained also in these terms. And essentially we have these two levels of interactions, one that is feed-forward, that cross, so to say, the whole visual pathway, and another set of connectivity that modulates these inputs. But we do not really understand many of these, so to say, the nature of this modulation. And a very nice and important example, in fact, is the connectivity from the LGN to the cortex, because we know that there is a massive back projection from the cortex back to the LGN. This represents, So the retina represents in the cat about 9% of the connectivity. So we do not understand exactly what the 91% is doing. This, I think, give in numbers also a clear picture that what we know about the visual system or the brain is we have far from a complete understanding of the processes. So here, an important finding that starts, so to say, exemplifies very well the notion that receptive field complexity increases. You can see here in these stimuli borders without luminance. This is what we call, and I'll explain, have another example, illusory contours, because they are induced by the other lines. So this is very interesting because you have a border without contrast, physical contrast, without light contrast. And in this study in the monkey, von der Heidt, he shows that in view 1, but in view 2, but not in view 1, you can have cells that are sensitive to these illusory borders. And this is very interesting. Of course, the responses are not so strong like the physical borders, but is an interesting discovery showing that receptive field properties can explain more psychophysical phenomena that is not so simple to understand, because of course this implies an interactions over space. So here another example of the increase in complexity in the response of the cells is the seminal work of Rob Desimone in '82, and essentially in view 4, responses are modulated by attention. I will not enter too much in detail because I just wanted to go showing exactly this concept that response properties increase in their complexity if you go through the visual system. The monkey here is fixates a point, and there is a way of asking him to pay attention to an object and to another object. And Desimone shows that responses in V4 are modulated when the monkey pays attention to one object inside the receptive fields. And this was, of course, extremely important because shows now internal process modulating the responses. So you need a more, how you say, broad account in order to explain perception. And this is, of course, not simply a representation of an image. Here, I brought one example for each visual area, or some of the visual areas. But this is a very important study of Nilsson in the 90s. Essentially, he recorded from an area that is known to be very sensitive to motion. You have many neurons where you can find very precise tuning to orientation, the direction of movement. And he used then these dots that could be completely random or completely coherent. And so he made a seminal work in manipulating the perception of the monkey by microstimulating the column he was recording from. And this, of course, was very important to the notion that the activity of single cells represents certain feature, because he could demonstrate, so to say, a change in the performance of the monkey if, for a particular orientation, if you stimulate this particular corresponding group of cells. So that's why this was a kind of a very important contribution at that time. And the notion then progressed with these findings that responses in the infratemporal cortex can be very selective. This is a work from the 70s, always characterizing responses, and you see that You can have a very selective responses to faces. Today we know that there is a very dedicated and complex circuit in the infratemporal cortex dedicated to face processing. This is the work of Winrich Freiwald. And this was the seminal work showing that cells in the temporal cortex can represent very complex stimuli like faces, and selectively, so they are not sensitive to other objects. So the notion here, as a kind of a global picture, was that receptive field properties could explain the way we link parts of objects and represent globals. For instance, here a screwdriver. This was the work of Tanaka in the 90s. And there is also very impressive findings in the amygdala from Rodrigo Quiroga, showing very selective responses to complex features as a particular face. You see, for instance, that the cell is very sensitive to the face of Bill Clinton, independent if this is a cartoon or not. So this was, in fact, an amazing finding, and we can discuss if this contributes or not to the concept that single cells response can represent in a hierarchical, convergent manner properties like whole faces. So in a sense, this concept, and I say this is a Cartesian concept, has been extremely successful because this very idea that the brain or the visual system can represent the world by their spiking activity has been, in fact, a very successful program. So the idea is that the ganglion cells can find better the contours in the image, so find better the objects, and the cortex you have a full representation of objects. So this is an extremely powerful and successful notion that the visual system represents by their spiking activity a visual scene. And this film, Bis aan de Welt, was by Wim Wenders in the 90s, was in fact very interesting in putting this idea clearly. And the film is a science fiction film. The idea was, the film says, okay, it would be possible then to build machines that could, so to say, read this representation. If this would be possible, it would be possible to, for us to have our dreams as little films. And the film is, in fact, very fascinating, the way he puts the images and also the whole story, because we get very addicted with these little devices, and people could not even eat anymore trying to understand their dreams in terms of images. But this notion is of we will be able to, so to say, read signals from the brain and to have this remapped into images. And if we have a representation of the visual world, in principle, it would be possible to have this reconstructed as small films. And amazing, it was, in a sense, possible 30 years later. And this is basically the work of Gallant, published in Current Biology in 2011, where he built a decoder using MRI, function MRI signals, and he could, so to say, reconstruct images from these very indirect brain signals, showing the power and how successful this Cartesian paradigm was. Because at the end, the very idea here is that it's only possible to reconstruct these images because somehow they are represented by the brain activity. But very simple phenomena remains still difficult to explain. And this is an example. So as I have shown, von der Height showed that V2 cells are sensitive to these borders where you do not have luminance, or the distortion of lines. So the lines are right, but you see them distorted. Why? Why it happens? Why it happens that we see that vision is a very, very active process. You see by this image that you always have something competing that is never, so to say, sleeping. And in a sense, this has to do not with the image, but the way we are built. And to try to understand this type of phenomena, what we call perceptual binding, was a very difficult problem all these years. So in a sense, we knew that studying single cells, of course, will be not enough to explain every phenomenon. And of course, the cells somehow they interact. But what was the nature of these interactions that could explain these perceptual phenomena? Technically, we call this visual binding. And biological motion is one example here. Shape from motion, you see shape from motion because these dots have coherent motion. But how you explain this? It would be not enough to just describe the properties of the response of single cells. Here, a very nice example that I love is the Le Merle, the Norman McLaren, a very simple animation, but it's very impressive because you get right away that this is a bird and the bird behaves like a bird. But the problem here is that you have a very, very, very few elements. So how we can explain the way we do this appropriate linking in a way that you perceive a single coherent object? And this has been a major question and difficult, so to say, to answer. The discovery of rhythms in the brain by new technologies where we could, so to say, record from the many cells at the same times, and basically the fact that there is a lot of rhythmic activity in responses has been a very useful framework in thinking how these interactions could be explained. And here is local field potential recorded from V1 in a behaving monkey. It's very clear that these responses of the population of cells are very organized in time. They oscillate like a sine wave in this case. I mean, if you do a power spectrum here, you have a very clear single peak. And this then has been a very an interesting possibility to explain interactions, because it has been demonstrated that when you have an oscillation, you have in the time domain very precise relationships across neurons, so across space. And this has been clearly demonstrated first in anesthetized cats, and has then triggered a series of experiments showing the importance of rhythms for linking or integrating activity over areas or over regions in the same area. This figure published recently in a small concept review of Buzaki, Logothetis, and Zinger shows very nice how oscillations are everywhere. So you have a comparison of different animals, from a mouse to a chimp to a human to a dog. You see that there is a conservation of oscillations in all these species. And for sure this is very meaningful, because this is not particular to a special system, and certainly it has a very important meaning in processing. Of course, you can ask, okay, at the end, oscillations are also defined by heuristically by their frequency bands, and we do not know if an alpha in a rat is really the same alpha in a cat or a human. There is a lot of debate, and I think that is far from being resolved. However, the figure also shows how common this order in the responses they are across the species. You would like to ask? No. So it changed then a little bit the scope of the experiments. Instead of just describing properties of cells, responses, also trying to understand how they interact in time. So the first very important method we used to describe these interactions in time was the simple correlation function, where you could then record spiking activities from many different cells and to track if they change their correlated activity as a function of the stimulus. So we always we use this principle of find correlates between features we can observe in the brain and perception. And here, this became a kind of a, how you say, icon of this paradigm, the Dalmatian dog that appeared in the book of David Moore, Vision. And everybody was, in fact, talking about the dog because it exemplifies very well the problem of visual binding, how we can link dots belonging to the dog and separated it from the background. And the hypothesis, the very simple was, I'm sorry, that cells having different, responding to different aspects of the scene. For instance, one cell responding here to the surface of the dog or to the dots that belongs to the dog will not synchronize with the cells responding to the background, and cells responding both to the surface of the dog, so that the dots that belongs to the dog will synchronize strongly their activity. So the notion here is that this binding or linking mechanism was provided by the synchronized activity across the neurons. And we have found a lot of evidence, and in fact, in many different levels of the visual system. Here is a cat during anesthesia. This is a cross autocorrelation function over time, and you see that there is a lot of structure in the response for the surface of an image that appears and disappears. And if you put an image that has no structure anymore, these oscillations are gone. So the basic idea here is this oscillatory activity can be used, so to say, to build the relationships we see at the perceptual level and explain phenomena like visual binding. Seminal work was this, published in '89, and this was in fact a kind of a how you say, very exciting discovery. A single bar could evoke or induce a strong oscillation in the visual cortex of the cat, but components that are independent, these two bars here, you have a flat cohelogram showing that there is no interactions. So this was a seminal in the sense that could explain exactly how the visual system links things. However, all these findings over the years, we have kind of negative findings. So in a sense, this became a very controversial. And here an example where you have two bars and one bar. So one bar meaning the cells responding all to the same object. Two bars, now they are separated in two ensembles by synchronized activity, so the cohelograms are flat. I will not go enter into much detail, but a few years later, or 10 years later, 20 years later, in fact, in this case, we could provide another explanation for this. So there was, and there is still, is a lot of debate and discussion on the meaning of of neuronal synchronization for feature binding. And important experiments were obtained in behaving monkeys. So these first experiments have been done in the anesthetized cat. Of course, in the case of the cat, it's difficult to explore more other aspects like attention and temporal expectancy. And the monkey, of course, the anesthetized cat cannot report in seeing objects as the monkey can. So this is the spiking activity. And there was, so to say, online autocorrelation functions computed when the monkey was seeing particular stimulus. And this is an example then of studies that have been done in the monkey now, non-anesthetized. And the monkey was reporting, so to say, seeing an object in another object. So it was a very straightforward test of the hypothesis of binding by synchronization. And you can see that there are very important negative results. I'm not entering into detail, but you have the possibility of tracking some of this work. Essentially, here, Palanca used a very nice paradigm where the monkey needs to report seeing one object or two objects in a way that in the receptive fields, the stimulus locally is always the same. It's just the contingency of the stimulus that changes. And the idea that if you have, so to say, linking, in this case, by the single object, you'll be very different in terms of the interactions of the cells as compared to two objects. And you see when he computes correlation or coherence for the local field potential or the multi-ion activity, these are essentially the same. So this was a very negative result in the sense that it was a careful study carried out, done in the monkey, where the monkey could really report what he was seeing. Another set of experiments, and this became, so to say, a different branch in the idea that timing is important in perception, came from the study of Fries in the NIH in 21. In fact, Pascal Fries came with a series of papers, and I would say even an explosion of papers, and it has been very successful in showing a link between selective attention and the phenomena of oscillation at different levels in the monkey brain, particularly an area, the area V4, that was known to be important for selective attention. So you see here the monkeys pay attention to different stimuli, and you can cue and say, monkey, pay attention now to this stimulus by changing the color of the fixation point, or pay attention to another stimulus, and you see that whenever he pays attention, there is an increase in the amplitude of the oscillation. And this shows that these cells are now more synchronized as a population, and this depends on attention. Here, another study of Taylor and Andreas Kreiter in 2005, a very nice study also, because he presented figures to the monkey, and the monkey had to match one figure and the morphing of another figure in the, so to say, other visual field. And whenever the monkey tried to see a form, there was an increase in synchronization. Here you have the two aspects, the aspect of expecting to see a form and seeing a form. It's not very clear if this depends on seeing a form or expecting to see a form, but you see clearly that, so to say, this contextual problem can explain the increase or decrease in synchronization. It's not only, in a sense, binding of features, but other aspects such as attention and temporal expectancy. And Pascal came with, Pascal Fries, in the work of Bosman, came with a very nice theory, and I would say important, that could explain selective attention in a way. I will explain. This has been recorded from 2 areas, V1 and V4, and he used two stimuli. So the monkey had to pay attention to one grating or another grating. And he recorded from area V4 and V1 at the same time. And it's interesting to see that the correlation of activity across V1 and V4 change as a function of the monkey pay attention to one object or another object. But there is a very specific and interesting finding here, is that the oscillation frequency change as a function of attention. So it was that attention was modulating a very important property for synchronizing, that is the frequency. So if the monkey was paying attention to this stimulus here, the 2 frequencies here in red for this stimulus, here in blue for this stimulus, they are different in V1. And and there is a very strong synchronization if he pays attention for this V1A. However, if he pays attention to V1B, the synchronization appears for this side, and for this side here there is no synchronization. So this is interesting because it shows that depending on what he is seeing, now the link feed-forward between V1 change with V4. And that this is very important in answering, in a sense, your question, because you show this study shows clearly that the linking or the functional connectivity is not explained simply by the overall structure of the brain, since it can be modulated by attention. So this is an example where the oscillation and the timing could explain this flexible or flexibility in perception without a need to change the structure of connectivity in the brain. We have found also evidence for this in monkey V1, in a study where the monkey was paying attention to one surface or another surface. So here these two are overlapped. It's interesting if the monkey was paying attention to the surface behind, there was an increase in frequency, and if he was paying attention to the surface above, there was a decrease in frequency. Not enter too much in detail. Is another example where oscillation frequency changes as a function of perception. So pay attention in 2 different objects. Different from Pascal, these objects overlap. And here I show data from a monkey also, in a paradigm where the monkey has to detect a change in the stimulus. And it's interesting, we found that at the end of the trial, when the monkey had to respond to the stimulus, there was an increase in oscillation. And then we started investigating this, and we could conclude that this depended on the expectancy of the monkey. So this is a little bit different from the idea of spatial attention, because you can expect things to happen in space, but also in time. And we used a paradigm where the monkey could expect a change in the stimulus at the beginning of the trial or at the end of the trial. So you can change what we call hazard function. So you expect always a change at the end, and there was an increase in gamma at the end of the trial. But when you change this, and the monkey had to expect always at the beginning of the trial, and time to time you put a change at the end of the trial, you could see an increase in gamma activity that depend exactly on the timing schedule of your stimulus. And this is, was, so to say, very interesting, because brings, so to say, a different dimension, not only selective attention, but temporal expectancy, as a process that can modulate gamma. So gamma may be important also for this very important aspect of perception or perceptual organization, because it's known that spatial attention and the temporal expectancy modulates the way we see. So this has been a very interesting finding that goes along with Fries and others finding on the role of gamma oscillations in selective attention. And I just would like to finish my presentation, that we have a little bit talk to discuss by showing another example we used to test how synchronization is important for perception. We used this paradigm where there is a figure that appears over in a point in time over the trial. In fact, the group of Charles Gilbert came 2 months ago with a very nice paper showing the importance of coherence in perception of Gestalt figures like this. And here we see, although the responses are the same, because the local features over the receptive fields are the same, there is a very strong increase in oscillation at the end of the trial. And this was not associated with seeing the figure. So it's an example also where the idea that oscillation is important for binding fails. However, there is this important notion that this is linked to the timing, because the monkey had a sense of the timing of the stimulus in the same way he had for the other tests we made. And so in a sense, it's a negative finding, and we contribute to the controversy. However, a very positive and interesting result, thinking in terms of temporal expectancy. And very impressive, depending on the flavor of the juice. So if the monkey was more or less motivated to cooperate, and particularly this monkey, he was very fond of banana juice. And they have an increase in the modulation in view one, depending on the reward size. So if we increase the quality of the reward, or increase really the size, have more or less motivation, and there was an important modulation of the gain of gamma. And this is very interesting, because show that even an area like V1, that is, so to say, a primary visual area, you can have these very large contextual effects that depends not only on the stimulus, but other aspects of the perceptual organization, such as the meaning of the stimulus, or such as expecting it in certain point in time or in space. Interesting also to point out that this here is for alpha and here is for gamma. There was always an increase in gamma and decrease in alpha, exactly as Pascal Fries has shown for temporal expectancy. Sorry, for selective attention. And here, just then to finish a last finding, there was a collaborative work we have made together, and we could show that depending on the phase relationship of the spikes in the ongoing field potential, the representation of the orientation could be optimized or could be better. It was the cells were, if you take these spikes that were, so to say, phase locked with a particular phase of the gamma cycle, these particular spikes could represent better the orientation selectivity of the cells, meaning that these phase relationships, so timing, may be important for the representation of the stimulus. And finally, a last slide showing that this may be important for explaining disease. And here, a paper of Peter Woodhouse showing that in schizophrenic patients there is a very clear decrease in gamma in a task where the patients were required to do a kind of visual binding for seeing these money images. And this is an important finding because it shows that, in fact, timing, in particular gamma process, may be important for us to understand the nature of diseases that involves interactions, large-scale interactions in the brain, like in the schizophrenia. Okay, that's it. I would like to moderate the discussion. I understand that I brought perhaps too many things for people that are not really used with this concept. However, we can come back and discuss more in detail some aspects of certain notions. And I would like to recapitulate. First, the idea of representation. I think that this is a very strong, so to say, concept, and that was very clear in the whole research program of the 60s and 70s and 80s and 90s, showing that basically we can understand how the responses extract features, in a way build internal representations by the neuronal activity. Who wants to comment on this? Who thinks that there is no representation in the brain, or it could be different? Yes.
Your question reminds me of a philosopher. I think he was in Harvard. The guy, Mind is Motion. What's the name of this guy? Van Gelder. Timothy Van Gelder? He was in Indiana. Okay, you know him. So he had this book that was very influential in the 90s, in which he argued that you could conceive of a brain that lacked representation. And the metaphor was very pedestrian, was a manometer. Basically, the measure of pressure would indicate there's an indicator of changes in pressure. And he says, well, you know, there's nothing being represented there. The fact that it's changing is what matters. And I think it doesn't survive 10 minutes of introspection. When people go into a coma, and they may stay in a coma for weeks, months, sometimes years, and they wake up, and they don't wake up properly themselves, but they wake up somewhat themselves, and they recover themselves. So there's no way you could imagine a brain that could do that, in my opinion, if there was no representation. So there is something that is latent, that is there, that does not need interaction of the environment, right? Otherwise you would sleep at night and you would forget who you are. But again, in philosophy, there's space for everything, right? So this argument actually thrived, and it was you know, the book is beautiful.
I think that an important point is what you do with the concept. And of course, particularly in vision neuroscience, we have, so to say, stretched the concept to a maximum. And I would like to put two things. One is why it became so successful. And I think that the work of Jack Gallant, showing that you can, so to say, extract information to reconstruct images, is really impressive. It was in this film of Wim Wenders as a kind of science fiction, and it's really amazing to see somehow it happening 30 years later. However, it's a concept that fails, because if you go a little bit out from the very crystalline visual maps of view 1, it's then very difficult, so to say, to extract information to build the image. So it's not that it's so clear like this. I think that is, and that this is, of course, a very important debate, because it somehow also guides where we should put our efforts or not. It became also very clear into the whole discussion of visual binding that there was an epistemological problem behind. Because if you have here an anesthetized cat, and you say, oh, he should synchronize these and should not synchronize these, how you can really tell how the cat in these conditions see and what should be bound or not. So you assume always that there is a kind of a visual world that is behind you, that is before you and here, independent of you. But it started becoming clear that is not really the case, that in fact these things are brought by the very interaction of the system with the visual world. So this David Marr idea that the visual system somehow represent features that somehow needs to be built is not necessarily the problem and or the most important problem.
So a couple of comments. One, respect to the reconstruction of the movies. This is not a generative process. So clearly there is information there, but the way he builds, Jack builds the models is based on priors on clips from YouTube, right? So, and then based on that, he chooses which clip is closer to the information that is present in the fMRI data, right? It's not generative in the sense that you take that data and you reconstruct the image. You use that as a prior to choose a clip. So I'm not saying this is cheating. It clearly shows that there is a huge amount of information there, but it's not straightforward as being able to completely reconstruct the image. That's one thing. And the other thing that I wanted to say is regarding to what Siddhartha was saying about Van Gelder. I mean, it's very clear that the problem is that, not the problem, that representation, perception is linked to behavior, and you cannot really untangle them. And that's what's been confounding the field, in my opinion, right? Because clearly, in particular, what transpired, for instance, to computer science is that there is this static representation of the world, and essentially if you go there somewhere in the cortex, you will essentially see a pixelated version of the world. And that's not the case, because that is always conditioned by behavior, by what is necessary for the organism to represent in order to act. So that's, that you can take that and say, okay, now there is no representation, it's all about action. But I think the truth is clearly that the two things are linked together.
It's also true that we are not really prepared, I would say even conceptually, to fully explore this relationship of action and vision. And for instance, when we came into this room, we have always this sense of a very, so to say, solid image and detailed. But in fact, if you think in terms of eye movements and what you could sample from this room here is extremely sparse. So there is in fact a gap from all we are discussing about vision and actually what perception is. And I think that if there is a message to have from what I talked, is that when we start thinking in terms of oscillations and temporal binding, there was an important paradigm shift, because we start thinking more in how the brain builds internal relationships and how this is related to perception, and not simply trying to understand the properties of how responses represent features. So not really understanding a more large-scale integration in the brain, because this was, so to say, more or less solved with this scheme of a fixed connectivity over a hierarchical feed-forward system. So I think that these are important notions, and if there is something new to be developed, of course, is the understanding of the nature of these interactions in a complex system like the brain.
I think these studies of decoding we mentioned Jack Gallant, but there are other people, a few other groups working on that. I think they are contributing major, these studies of decoding by Jack Gallant and other groups are contributing a great deal of new information. So we usually tend to talk about the fact that it's amazing that you can decode stuff from visual areas. But I think that there's one recent publication in Nature Neuroscience by this group 2 years ago, I think, which is, the title talks about attentional warping of the representation. And I think if it is true, if it's replicated by other groups, etc., I think it's remarkable, because what they do is they image people as they watch an entire movie. And then they score each frame of the movie according to its semantic characteristics. So if you see an airplane and grass, the raters will say there's an airplane and the grass. So you end up with a vector of labels for each frame. And then what they do is they look for how each voxel across the entire cortical mantle, how each voxel tunes to this particular feature, say vehicles or tools or people. Okay? What is amazing is how distributed it is. So you have voxels that tune to any category throughout the cortex. So you don't have little, it's the opposite of this idea in the 80s, in which these tools are here, in the right bank of the superior temporal gyres. This is gone in this new representation. And what is most amazing is he does the experiment by having people watching it passively, and then he repeats it and says, okay, now you're going to watch the same movie, but you will only pay attention to people. Forget about vehicles. And then the entire map tunes to people, to vehicles. The entire map warps in the direction of the attention. And then the things that are associated with the target category also get expanded. So if you're tuning for vehicles, you also tune for tools, you tune for inanimate things, which goes back to ideas in math and philosophy 100 years ago, and Guillermo is the person to talk about this, that concepts can only be represented in relation to other concepts. You only know what a dog is if you know what a cat is, that's to simplify. And the fact that you can recover those maps and see this big shift in tuning, I think it's a whole new world. It's not the way we used to think the brain 10 years or 15 years ago.
But this shows very clearly that, in fact, the visual world is not there, it's defined by your relation. Jorge Nascimento, he showed to me a little video of a cat chasing a spot in an iPad, and it's really amazing to see how he's always running to this thing in the iPad and interacting to it. Now, if you show a movie to this cat, you'll be completely out of his cognitive space, and you'll be much less interesting for him. So I think that if this idea of taking for granted, and this is of course very linked to the concept of representation, that we have an image that somehow needs to be, how we say, parsed in segments, and somehow the visual system then reconstruct these and represent as a concept in the brain, is not necessarily, so to say, the good way of thinking the problem. And interestingly, if you think in terms of gamma synchronization, if you, so to say, review the papers that have been, so to say, how you say, not denied, but how you say this, that, not rejected, but that has a kind of a problem with another paper. How you say this? That enter in a debate or a controversy, they most are linked to the idea of binding of features. And I would say at least three. This was Castelo Branco paper in the anesthetized cat, and the paper of Chiller and Stoner in the monkey. Clearly, they show that this is not the case, that synchronization really explains segmentation of the plaid stimulus. Another is the one I have shown to you, Palanca and Andreas Kreiter. And there is another series of... and why? Probably because the concept that you have a representation of features and somehow this needs to be linked is not necessarily a good concept. Interestingly, if you see the whole idea of linking gamma and attention and temporal expectancy, this is a major growing field or evidence. And I find that this is interesting because it also has to do with two different concepts. One that is still very linked to the idea of understanding how the brain extract features and represent as global objects, and the other that starts from another perspective, just trying to understand how relations are built in the system, and this has to do with internal process such as attention, emotion, temporal expectancy, learning, memory. So it's interesting because the philosophy can also time to time guide. And I think that this is a good example. I have a question to Siddhartha now.
I was wondering about papers that treat representation as a multisensory kind of search, like in schizophrenia, when you showed that paper, maybe gamma representation for visual representation are impaired, but they are essentially auditorial hallucinating people. So kind of sometimes the representation are not visual, but they could be in a multisensory knowledge for that person, semantically speaking. The semantic of that concept could raise as another sensory field. Is there any paper talking about that kind of representation in a multisensory approach?
Not really that I know. If you could explain the lack of synchronization because people are not unable to, so to say, link visually, but they could do in another way. However, I think that this idea of we could have internal representations during hallucination, also during dreaming. And my question to Siddhartha is, there are differences between your dream images and the images you have from your interactions with the world. Do you think that we could apply the same principles of organization to explain, so to say, visual binding during dreaming in the same way as we have in our daily interactions with the world?
Well, I probably should just say I don't know. We don't know. But I have no reason to believe it's a different process. The thing is that when you're seeing things during sleep, during dreaming, we'll talk about it the next session, you're doing it with no noradrenaline or serotonin in the system, for example. And that makes a huge difference in how neurons respond. Could you guys hear me in the back? So the fact that the visual images during dreaming are so faint is not, I don't think it's because the rules of binding, if those actually exist, the mechanisms of binding, I don't think that it's because they're different. It's just because the neurochemical environment is so different. So when you get image, so I'm going to talk a little bit. I have one slide about decoding during dreaming. In fact, it's not REM sleep, but it's the N1 phase. This Japanese group, Horikawa and Kamitani and other guys. And in fact, if we think that the Jack Gallant images in V1 are course. I mean, what these guys are saying, this is decoding of dreaming, is actually a lot of wishful thinking with a good p-value and a paper in Science. But it's nothing you could. I mean, when you see those movies, many times you see it's a person, for example, and it's a person in the right part of the screen. So it's pretty good. You don't know which person it is, but you know it's a person, not an airplane. With dreaming, the signal is much worse. We met Jack Gallant in a meeting in Tennessee a couple of years ago, and we talked about this, and he said, oh, I actually tried to do it with dreaming, and he showed us the movies, and it's basically white noise. and so much so that he didn't publish it yet. And he said, oh, this is Gallant. So I don't think that the rules will change just because we're sleeping. I think, you know, the rules are the same, but what is available at the level of each neuron is very different. So if a neuron has a lot of noradrenaline supplied to it, you know, typically in different parts of the brain, you'll say, well, the signal-to-noise ratio will increase. You know, neurons will respond in a stronger manner to stimuli, and other neurons will shut down. So in sleep, it's the opposite. Everybody is sort of free to respond, and this may explain why things are so hyper-associative. But there's a lot of hand waving here because we don't know.
I have a specific question about the oscillation, the frequency of oscillation. Besides gamma and alpha oscillation, is there any other band of frequency like beta oscillation? Any evidence for this kind of binding process and attentional process? Is there any evidence for the beta, for example?
Of course, the oscillation is defined by the frequency band. The important question here is these different frequency bands mean always different process. And the first thing I would like to tell you is that individuals have very different frequencies. And this was a little bit unexpected, because when we start investigating this, particularly in the anesthetized monkey, frequencies were always around 40 hertz. And in the 90s, we are talking about 40 hertz always. And the 40 hertz was, in fact, synonymous of gamma activity. And this is clearly, so to say, faster than beta, and clearly faster than alpha. There was a particular mu rhythm that was a kind of around 14 hertz described in the cat, but there was a notion of a golden frequency, and 40 hertz was this frequency. However, when we start investigating V1 of the monkey, it became clear that there are many different frequencies, and some monkeys could have frequencies as fast as 80 hertz. And this is, means twice in terms of frequency. This is a problem. Why? Because if you think that the frequency carriers, so to say, a phase space where you can synchronize, it means that this phase space here is much shorter because he's too fast. And as compared to a monkey that does it, so to say. And we then, in our particular group in Frankfurt 8 years ago, or 7 years ago, we made a kind of unexpected discovery that the rhythms, if we compared the hemispheres, they were the same. And across the individuals, they were clearly different. Of course, this suggests that rhythms may be linked to the structure, because the hemispheres tend to be the same, and the individuals tend to be different. This can be due to the ontogenetic, so to say, history of the individuals, but could be also due to the genetics. Pascal Fries then came with an important finding comparing twins, and you see that identical twins, they have identical rhythms, and if they are different, they have different rhythms. So I think that is unclear until now if why these important differences in frequencies. The paper of Pascal also showing that the frequency can change as a function of attention is also very important. I understand that you ask about so the bands, but the point here is that this is a kind of a loose definition. For instance, in the New World monkey, the capuchin monkey, Pedro Maldonado described the same phenomena as in the macaque monkey, but for a beta frequency, not a gamma frequency. This is meaningful or not? So I think that this is extremely an open question, but of course very important. And also has to do with this not knowing really if we are talking about the same process, because we do not fully understanding these differences. Yes.
For humans, is there an experiment with attentional processing and the beta synchronization, maybe?
There is. In fact, when we came with this idea showing that the structure may be very important in explaining frequency, the group of Pascal in a paper of Van Peltz last year, he, so to say, shows that we are wrong. They show that we are wrong, saying that the frequency in the human being can change not as a function of eccentricity, so related to structure, but as a function of attention. Depending on selective attention, frequency could change. But these changes are not beta gamma, but so to say about 7 hertz. Still important in terms of how we synchronize, because to synchronize signals that have different frequencies is more difficult, so to say. Yes.
Thank you for your talk. It was a nice talk. And I have a question. I'm an elf, beginning my communication with hobbits. And I'm always listening. You're talking about activity, the pattern of activity, and alpha and gamma. And you know nothing, or you know something about the connectivity. If someone tried to understand the relationship between the connectivity and the perception, or something like this.
You say connectivity and the rhythms.
No. The connectivity between different areas of the brain, the structure, the network.
Yeah, there is now. In fact, I would say a massive interest on this, because people really start understanding that without understanding the connectome, you cannot understand function. And there was this, how you say, extremely useful technique of being possible to extract this information from the MRI imaging. So there is, of course, a lot of interest on bridging this, so to say, different data. Now, if we have a global picture of how these things are, so to say, connected, so the connectome and the rhythms, I think that we are far from this. We have been trying to make an effort in this sense. What's your name? Rafaela. We had, and your name? Garcia. We had a very interesting situation in the lab. We have 3 monkeys. One monkey that was Nico oscillated at 70 hertz, Lily at 40, and the Pepe, we have never seen a gamma oscillation from him. So, of course, we were completely puzzled. And we thought, why Pepe is different? Why we cannot see these strong gamma limit cycle oscillations in his visual cortex? And I was very worried, and I tried many times. Oh, we are not in V1, we're probably in V3, but we were in V1. And we were thinking if we could track then differences in the microscopic organization of connectivity in V1. This is a delicate project because it needs, so to say, so to say, to sacrifice the monkey in order to study anatomy. So until now we were not really willing to do this, but it's known, so to say, in the background conversation that gamma sometimes may be missing. And if you see even, so to say, very solid features like orientation selectivity, so these maps I have shown, they are not always the same for every individual. There are a lot of variability. So it's amazing how it's possible to have the same functioning so that the perceptual skills of these animals or these individuals, human beings also, are about the same, but there is clear differences in organization also at this level. And it remains still a puzzle. So we hope that we can understand it better in the future. But of course, this is a complex and a difficult answer, because it implies in bridging data from different worlds, the anatomy, biochemistry, and physiology. So we hope that to have better mathematics to describe our phenomenon. This is also important. So most of what I have shown to you comes from a very classical approach of how to say, hitting the system with a very particular stimulus in very unnatural conditions. How much we have biased, so to say, our observations, trying to find this beautiful phenomena of oscillations, and in a sense we are somehow going to a very unnatural situation. Yes.
If you record from a bird's brain or from a reptile brain, you don't have these oscillations. You certainly, in the bird brain, you certainly don't have any of that. In the reptile brain, it's not so clear. It does have a laminar structure. A lot of the oscillations have to do with the fact that the neurons are stacked in an organized manner according to those columns. But this is not a general feature of the organization of the brain. This is a mammalian feature. So if we had a theory for binding that depended on gamma oscillations, then you wouldn't be able to explain birds. So there must be something to be generalized, and this is the business of elves, that it goes beyond particular signatures of activity that actually we know from the brains of, a couple of species in monkeys and, some species in rodents and cats. What do you think of that, Sergio? Well, right, so it could be an epiphenomenon and it could be totally unrelated, yeah.
Siddhartha, to link the idea that, I'm trying to find this figure here, but to link the idea of gamma oscillation, the columnar organization of V1, this is half true, I would say. What is important, in fact, first, is that we have different rhythms. This is very clear. To say that the birds do not have rhythms, I cannot really agree, because have different rhythms. So the important question is to know if these different rhythms, they serve for the same role or not. For instance, in the optic tecton of the pigeon, when I made my PhD thesis in Paris, we found this in the optic tecton of the awake pigeon, and they were very strong. So it's not that you do not have rhythms in birds, but it's sure that the birds are very different in the organization, and I agree that all these things may be linked and the column may be important, but simply saying this is because of column organization, this will be wrong. However, does gamma in the visual cortex of the cat is exactly the gamma we are talking about in the human visual cortex? Not necessarily. And this is a problem because at the end, the definitions are always by the bands. So it's a priori definition. But not necessarily we have differences or different mechanisms that you could understand that this alpha is different from gamma, because this said is interesting to mention that it seems that gamma is related to more local processes. And the more slow frequencies like theta and alpha is related to more global process. And we have been very frustrated over years not to find really strong coherence for this very strong local synchronization in V1 of the monkey. And perhaps the reason for this is that gamma is very local. It means that synchronizing across a distance of 10 degrees is very global. But okay, in some sense also you needed to respect your negative result, because it means it has a meaning. So I think that the field is still, how you say, these are, it's unresolved, it's not clear.
It's 10:13, so we started in time, we'll finish in time, 10:15. Ask a big hand to Sergio. There should be coffee and something edible outside.